and the trees give a home to the fleas in my hair. A home for fleas, a hive for the buzzing bees, nest for birds, there ain't no words for the beauty.
In case you have never heard that song, that is the theme from the 1970s hippie musical Hair, which I thought might be appropriate for this chapter. Okay, so this is part 2. So now we're going to talk about the life cycle of hair. Life cycle of hair consists of 3 distinct stages. And a way to help you remember this is to remember the acronym ACT, ACT. Anagen stage is first, catagen stage is second, telogen stage is third. So the anagen stage lasts the longest. This is a period of active growth. Catagen stage only accounts for about 2% of hair growth, and the telogen stage, this is where the hair follicle is dormant. So this is the point where hair is no longer growing, and this is where the hairs will be easily shed. So it's the hairs in this stage that you find in your shower, that you find stuck to your coat, and various other places where you shed hair. So these could easily be left unwittingly at a crime scene. So here's some pictures of treated hair, and we'll look at some treated hair under the microscope when we look at different types of hair. So when hair's been dyed, what you'll see is color distributed throughout the hair, so cuticle, cortex, and medulla. If the hair's been bleached, it has a yellowish tint, and what bleach does is remove the pigment, so you won't see any pigment granules. As hair grows, of course, the roots come in and the natural color returns, which keeps the colorists in business. So rate of growth is about 1.3 centimeters per month. So knowing that, if an entire hair is found that's been color treated, we can estimate when the hair was last color treated. So all we have to do is some simple division. You measure the length of the hair that's naturally colored, so that would be the root, the regrowth. So you measure the regrowth, you divide by 1.3 centimeters. So that's going to tell you how many months since the hair was colored, since the hair grows at 1.3 centimeters a month, approximately. Little different for everybody, but that's an approximation. Okay, so Jessica's got some roots here. So let's say the root region measures 3.2 centimeters. So what I'd like you to do is calculate the time since the hair was last color treated. So let's go back. Remember, you're going to take that 3.2 centimeters, which is the length of the hair that's naturally colored, and divide by 1.3. Easy peasy, simple division. So give me that answer and we'll go over it in class. Another way to look at different types of hair is to look at racial differences amongst hair. And the 3 major races of hair that have been delineated are African, Asian, and Caucasian. If you're Hispanic, you're kind of a mixture of African and Caucasian. So let's look at African hair first. Generally tends to be curly, very dense pigment but unevenly distributed. And the diameter of the shaft varies. One thing that we see, this is the cross-section of the hair. So this is a picture of hair if it was actually sort of sliced up. And you can see that it looks sort of flattened. And of course, here's the hair is curly. The medulla can either be fragmented or absent. Okay, next is Asian. Asian hair tends to be very coarse and straight. Diameters doesn't vary too much, and again, unevenly distributed pigment. Usually a continuous medulla, and cross-section is more round. Caucasian can be straight or wavy, evenly distributed, fine pigment, and the diameter of the shaft is moderate, and the cross-section of the hair tends to be more oval. So the problem with identifying hair as to race is that not a lot of people are just one race. So often, you can't really see definition in the traits. Individuals who are of mixed heritage will sometimes exhibit properties of all of the heritage, and so you're not really going to be able to classify it very well. And we will look at hair of different races in class and see if we can figure it out. So when hair evidence is collected, the best way to do it is to try and get one hair at a time. If it's just an isolated piece of hair, you can use tweezers. Otherwise, if it's more than that, you want to use a lint roller or some sticky tape. And you can use a special evidence collecting vacuum that has a brand new filter when you start collecting the evidence. And when you have a lot of hairs in a large area like carpet and bedding, this will often work well. But you want to try to collect them individually if you can, particularly if there's not a lot. And the hair will go in paper packets. And if you collect with sticky tape or a lint roller, you can just pack the whole thing in a plastic bag. If the hair is stuck to an item, if possible, you can put that item right in the plastic bag. And when you're collecting hair from a suspect or a victim or somebody else you need to rule out from the crime scene, you take from the pertinent regions of the body, depending on what the crime was, 50 head hairs. If there was a sexual assault involved, then you would take pubic hairs, and you would take about 25. And you want to take them with the root intact if possible. So those samples would be called control samples. So again, it's from anybody who might have left evidence at the crime scene. And these hairs will provide a baseline for comparison. And it's important to collect this many simply because not all hairs are the same. You look at a person who has gray hair at the temples and dark hair on the side of the head, those hairs are going to look different under the microscope. So you need to get a representation, which is why in a crime lab setting you would look at a lot more hairs than what we're going to do in class. So first thing you want to look at macroscopic characteristics, the length of the hair, the color, whether it's curly or not. Then microscopically, what's the medulla pattern, one of those 5. The pigmentation of the cortex, what type of scales on the cuticle. You want to make sure that you separate human from animal hair. And if it is animal hair, see if you can figure out the species. Try to classify racial origin of the hair. And this can be used to either link a suspect to a crime scene, or it can exclude a suspect from a crime scene. Here's a picture of a scanning electron microscope that can be used in some hair analysis. So one of the things that hair is particularly good for is to measure either ingestion of drugs or poisoning. When you ingest a drug or a poison, it enters your bloodstream and then it breaks down into certain metabolites. So because the hair follicle has a blood supply and these metabolites get into your blood, the hair actually absorbs these metabolized drugs that are fed to the follicle right through the bloodstream. So for somebody like me who has long hair, if there were poisoning, this hair would represent a certain time period of growth. If there were poisoning that happened, it would still be present in the ends of my hair, and you would see it until exposure to the drug is ceased. So you could possibly say, okay, right here at this point, we could figure out that there's poison in this part of the hair, there's no poison here. We could determine that this person was last poisoned or last took drugs a certain amount of time ago by looking at the hair growth. Now, of course, this only works if someone has long hair. Someone who shaves their hair or has a buzz cut, you're not really going to get too much out of that. So hair is actually a better indicator of long-term drug use than a urine specimen, because a lot of drugs metabolize out of the urine within a couple of days, but hair will show drugs for a long time. So that can really tell what someone's been doing or whether they've been poisoned. Another test that's done, and this is not a routine test that's done in many labs because you have to have a nuclear reactor to do it, but it does have some value. A neutron activation analysis, basically what that does is look for various elements in hair. And what happens is the hair, and it can be as little as a 2-centimeter strand of hair, so it doesn't need to be that much. You put the hair in a nuclear reactor, it gets bombarded with neutrons, and the different elements that are in the hair will give off different signals of radiation. So you can identify elements like argon, copper, silver, sodium, zinc. And because everybody will have a unique combination of these elements in their hair based on their exposure, the probability of 2 individuals having the exact same concentration of 9 different ones is about 1 in a million. So, even 2 people who live together, they go to different jobs, they eat different foods, drink different things, maybe take certain vitamin supplements or medications. So all of these things that we do and that we're exposed to are going to affect the element composition of our hair. So that's why it's kind of like DNA in a sense. And there is a case study that we'll be talking about in class in your textbook. So DNA analysis, of course, is only possible if you have the root at the base of the hair, or the follicle, or the bulb, which is another term for it. So you can get that when there's been a struggle and hair has been torn out. And so if we look here, this kind of looks like a match head here. This is the follicle intact. So this is hair that's been removed and the follicle's intact. Here, the hair was pulled out and the follicle got stretched and pulled a little bit. But we would still get usable DNA off of that. So the DNA analysis of the hair, when we talk about DNA analysis, we usually take it for granted that we mean nuclear DNA and not mitochondrial DNA, although we can get mitochondrial DNA from hair. So what's the difference? Nuclear DNA is unique to each individual because the nuclear DNA, that's the DNA that's in the nucleus of all of your cells, and there's no one who has the same DNA as you unless, of course, you have an identical twin. In certain cases, when a sample's old or degraded, sometimes you may not get nuclear DNA, sometimes you may get mitochondrial DNA, or you may use mitochondrial DNA for a missing persons case if you have DNA from family members. Mitochondrial DNA is the DNA that's present in the mitochondria of our cells, hence the name mitochondrial DNA. And because the original cell that you started from was an egg cell that had its own mitochondria, all of your cells have this same mitochondria that your original egg cell did, which is the mitochondrial DNA from your mother. So you and all of your siblings with the same mother would have the same mitochondrial DNA. So we can use that in a missing person's case. Maybe someone goes missing, we don't have a DNA sample on them. There's a body found. If we can get mitochondrial DNA from siblings or a mother, then we may be able to determine whether this is the person that we're looking for. Of course, if you do have nuclear DNA and you have a sample to compare it to, then you can get individualization and you can identify somebody this way, whether you're identifying a missing person to a body or whether you're identifying a criminal. So that's it for hair analysis. See you in class.